Hello and welcome to Science in Plain English, the 2017 edition. Thank you very much for coming. I'm Roxanne Bogutska, and I am the founder and convener of the UT Science Communication Interest Group, and I welcome you all here to practice something that you will need throughout your careers, which is to be able to talk about your science to anybody, not just your scientific peers, but to the woman on the street, the person on the city council, the op-ed for your newspaper, people who are not involved in the scientific endeavor but need to understand how important is the work that you do and what it means for our society. So thanks for coming out here. I want to introduce our panel of judges. We have Julie Burwald, who is a science author and writer of the forthcoming book Spineless, The Science of Jellyfish and the Art of Growing a Backbone. Be looking for that in November. And then we have the Director of Communications from the College of Natural Sciences, Christine Sinatra, who is here. And last but not least, we have Nora Ashura, who last year in 2016 was the winner of the Science in Plain English contest. And I'm going to ask her to say just a couple of things about that experience.
Oh, hey, thanks. Unlike last year, this is not attached to a stand, so I don't have to feel victimized by the height. So I won this competition last year, and with the gracious contributions of the ARA, I was able to go to the AAAS annual meeting in Boston, and it was amazing. I feel like when I participated in the competition, my primary goal was figuring out a way to explain what I felt was really complicated in a way that wasn't just simple but also entertaining to actually get you interested in what I was doing, because talking about cancer and Star Wars might be a little bit more alluring than talking about a metabolite called kynurenine and how it interacts with T cells. So I think that after I had gone to the conference, it became even more clear why that was important, because in order to combat misinformation about science and what we do as scientists, it's really important that we're clear about what we do and getting that back to the public. And there's obviously a disconnect, and I feel like a lot of scientists in academia feel that right now, that there's a disconnect between what we do and what's being portrayed in the media and in the public's eye and how important it is. So best case scenario, you come up here and you win, which would be great. But at the very least, you're taking really necessary steps to hone your craft. And if you remain in science or not, it's really important that you get this experience and you become more competent in explaining really complicated science, especially if you stay in science. So I thought it was great. I hope you think it's great, and I'm really excited to hear y'all's speeches.
Thank you very much. Okay, so yay, thanks. Okay, so this is how it goes if you haven't been here before. You will have up to 3 minutes, those of you who are contestants, you will have up to 3 minutes to talk about your science. Tell us your science story. I have a really obnoxious web-based egg timer that will make a really obnoxious noise at the 3-minute mark. So you will be stopped immediately upon 3 minutes. The audience will then have 1 minute to vote. You are also part of the audience. So you have an orange ballot and a pencil in your hand, then you can and should vote on the folks who are participating. And here's what you see on your ballot. And how you can think about assigning a grade to each participant. You'll notice that the contestants are all wearing really large numbers like they're in a marathon. And I know sometimes public speaking can feel like you're in a marathon, but just kind of take a good slow breath and it's going to be fine. You're all going to do great. Okay, so you vote by the number. We're not going to be using names during the voting period. So look for the number on your ballot, and then that's where off to the right you'll be writing the grade. Okay, so without further ado, I guess we probably should get started with contestant. Let's start with contestant number 1. How about that? I'll give you the go sign. Does anyone need a ballot? Does anyone not have a ballot? Oh, well, let's fix that. Okay, they're back here. They're back. Oh, okay, great. All right, just hang on just a second while I get this set up. Please stand by. See, I couldn't follow the picture of the top which screens were which, so that's why
We good to go?
we're working on the screens here, so sorry for that.
It's okay.
Want to pace a little bit or something while we work on this.
I'm good for that, actually.
We didn't want it at first, but we want it now. Okay. All right. I'm going to go ahead and say, go ahead and just please get started, contestant number 1.
All right, sounds good.
Now.
According to the internet, there are exactly 109 uses of the horseradish plant. And I'm here to tell you today about the 110th as a way to detect breast cancer early so we can stop it in its tracks. Hi, my name is Abhi Rao, and I'm working with a protein found in horseradish called HRP. Scientists find this protein really interesting because it serves as a reporter,
Don't worry about it.
which means that it tells us, hey, look, there's something right here, by lighting up in a way that scientists can measure. Now to introduce the actual hero of my research project, the aptamer. The aptamer is essentially a really small piece of biological Velcro, if you will, that attaches to something, HRP in my case. I'm looking for an aptamer for HRP in order to develop an efficient test for a certain type of breast cancer. This test focuses on a certain kind of breast cancer known as HER2 positive. In about 25% of breast cancers, a protein called HER2 goes on a multiplying rampage, causing certain doom for the cells around it and causing them to become cancerous. The good news, however, is that researchers have already developed an aptamer for HER2, so my job is pretty straightforward. I will take my HRP aptamer, the one I'm trying to find in my research, combine it with the HER2 aptamer, the one that's already been found, on one side, and combine the HRP, that's the light up protein, to the other. That way, every HER2 is connected directly to an HRP that'll light up and say, hey, look, we have some HER2 over here. So how is this crazy guy up here talking about horseradish and how cool it is able to find this aptamer? The answer to that question is a process called Select, a game of pick and choose. First, I'll introduce a bunch of aptamer potential candidates to HRP, find the ones that stick, throw out the ones that don't, make copies of the one that stick, and keep doing that for a couple more times. The goal is, by the end of this, that I will find an aptamer for HRP that I can use as a test. So whenever there's a lot of HER2 present in a potential cancer cell, HRP will light up a lot, and clinicians will be able to make a better decision about how to treat this patient. All right, so thank you, I appreciate it. I'd like to thank Dr. Gwen Stovall, the UTFRI program, as well for their help in my project. I appreciate it.
Okay, you have 1 minute to vote. Anyone still voting? Everyone done voting? If so, we can have speaker number 2.
How tall is this? Okay.
And ready? Begin.
Hello, I'm Kirsten, and I am an undergraduate researcher in a neuroscience lab under the Center for Learning and Memory. This research is critical because it helps us understand how we can encode, retain, and retrieve information really efficiently. And I think as students, we all want to know how to utilize that. So my lab does this by performing 3 steps: what, where, and how. First, we try and understand what patterns we can observe in participants in behavioral tasks, which are usually computerized games, as we call it, to make it fun. And we take these games and we have them perform some sort of task that's going to give us information about how they learn and how they store information. So if we want to learn about categorical learning and how they can recall detail, we'll give them a task of maintaining a storage of paintings and the corresponding artists and seeing how they can recall the artists 1 week later. Second, we use the where, and we want to understand where the brain activity is localized. So just like an office space can have a cubicle devoted to Peter in accounting and a level of its own for the boss, our brains have specialized areas for each of the functions that it's going to be performing. And we want to understand where each of these functions are located. And this is done using functional magnetic resonance imaging, or fMRI scans. This takes the distinction between fresh blood that's coming to our brain and the used blood that's depleted of nutrients that our brain needs in order to detect what areas are active by using magnets. Okay, I'm going too far. Okay. So we use magnets in order to generate a response from this blood, and we get a distinct response from fresh blood versus used blood. And that way we can tell what areas are active and reconstruct a 3D map of the brain. Finally, we want to understand how these things come together, how we can take the patterns that we observe in a behavioral task and the activity that we can see by throwing images at the participants while they're in the MRI machine, and understanding the activity that occurs along with these patterns to localize what is occurring in our brain. So in summary, we take the what, where, and the how, and we use it to create a map of our brains and understand where learning and memory can fundamentally come from. Okay, thank you.
Let's take 30 seconds to vote. Speaker number 3, head to the front. Just hang on just a sec.
Okay.
Okay.
Resilience is one of the most important traits that a species can have in the natural world. My name is Savannah Troy, and I study the mountain white-crowned sparrow, or white-crowns for short, and the many interesting ways in which these little birds are surprisingly tough despite how fragile they seem. Mountain white-crowned sparrows are about the size and weight of half of a banana. They can fit very nicely into the palm of your hand and are very breakable. But at the same time, they migrate every summer to breed in the high elevation environments in the Rocky Mountains and the Sierra Nevada mountains. There are three main challenges that the birds face in environments like these. First of all, the breeding season that they have in summers there is very short. There are only about 9 weeks available to the birds to arrive after migration, to mate and establish territories, to raise their young, and then gain enough weight and eat everything that they can so that they can survive migrating back to their fall wintering grounds. Second of all, there is an intense amount of competition in these environments, both between white crowns and between white crowns and other species, to establish a territory where there's enough resources to build a nest and successfully raise the young. Finally, the environment at such a high elevation, especially in the mountains, is really unforgiving. The temperatures vary widely. It can be below freezing at night and then above 80 the next day. And the storms are violent and unpredictable. And the white crowns have to survive all of this in their tiny open nests that they build on the ground beneath willow bushes. As if that wasn't enough challenge for these birds to survive, they also have some of the world's neediest babies. White-crowned sparrow babies have a much higher metabolic rate than most of the birds of similar species. As a result, the parents have to balance their time between gathering food in this environment where resources are scarce, both to feed themselves and feed their babies. But then at the same time, young white crowns don't develop a lot of body regulation processes, like being able to regulate their own temperature, until after they leave the nest. This means that the parents also have to consider how much time they need to spend sitting on the nest to keep the temperature at a nice, safe level. Bearing all of this in mind, the most exciting part is that researchers up until this point haven't really looked at how white crowns balance their time in this environment, especially when they have the nest to consider. So what I want to look into is the ways in which the ambient temperature and all of the wide fluctuations that it can make from 20 degrees to 80 degrees in one day impacts the way that parents spend their time. Thank you.
30 seconds to vote. And now let's have speaker number 4. Ready? Go.
Do you remember the last time you renewed your driver's license? If the answer is yes, you were probably asked if you wanted to become an organ donor. And if you agreed, you have a little red heart on your license that identifies you. But unfortunately, more than 60% of these donated organs are thrown away each year. This is because we have no way to store them. We can only keep them for 8 to 12 hours. We can't just freeze them due to ice formation. The reason that ice cubes float in your drink is because when water turns into ice, it expands and forms ice crystals. Inside cells, these new sharp ice crystals pierce through the cell membranes, and they kill the tissues that make up your organs. So if we would try to freeze organs, they would die, and we wouldn't be able to use them to save patients anymore. I study chemicals called cryoprotectants, and these work by forming bonds with water, and they make it unable to form ice crystals. The cryoprotectant that I use is called DMSO, but unfortunately, it has a good side and a bad side. While DMSO prevents ice formation, it's also toxic to cells, and it can hurt other things inside living systems. This is why we're studying a new chemical called cryoprotectant toxicity neutralizers, and these do exactly what they sound like. They interact with cryoprotectants like DMSO and make it less toxic. They make it less dangerous to living systems while continuing to stop ice formation. So if we tried to study organs all at once, we would have no idea what was going on. They're way too big and way too complicated. Thankfully, we can use proteins as model systems to understand what's happening. The protein that I use is called lysozyme, and ours is purified from egg whites, just like the ones you eat for breakfast in the morning. So we can use this protein as a model system to understand how we can preserve larger things. The way we do this is by looking at the melting temperature of a protein, or when it unfolds, breaks down, and stops being usable. We can look at the melting temperature of this protein in different mixtures of our cryoprotectant, DMSO, and our toxicity neutralizer. The way that I do this is through something called spectroscopy, which basically uses a very powerful beam of light to give us structural information about samples. The first thing I did was spectroscopy on my protein in increasing concentrations of DMSO, which is our cryoprotectant. And we saw a decrease in the melting temperature. This makes sense because DMSO is toxic, so it's hurting the protein and making it less stable. So it's going to melt faster than a regular protein. However, when we added our toxicity neutralizer to this mixture, we saw an increase in the melting temperature. This is good. It means that the neutralizer interacted with DMSO, made it less toxic, and helped stabilize the protein. So one day in the future, we hope to be able to use our toxicity neutralizers and our cryoprotectants together to preserve living systems, and hopefully reduce the size of the organ wait list. I wanted to thank my PI, Dr. Baez, and my mentor. Thank you.
Thirty seconds to vote. Now, if we could have speaker number 5. Say when. You ready? You ready?
Yeah.
Go.
So today I'm going to talk to you guys about the field of mass spectrometry and a very recent invention in that field called the mass spec pen. So if you guys don't know what mass spectrometry is, it's basically the Sherlock Holmes of chemistry. Like Sherlock Holmes, it can take very small amounts of evidence and make incredible conclusions. So what mass spectrometry does, it takes very small samples of chemicals and it looks at them and you can build a lot of information from that. So the reason why you can apply it to human tissue is because we are made of chemicals. We are made of chemicals such as lipids or fats, proteins, and also sugars. And because the sugars, proteins, and the fats in tumors and cancer and in normal tissue are different, mass spectrometry can basically look at the chemicals and tell the difference. What the mass spec pen does is it's basically the mass spectrometer's magnifying glass. It's a detective's magnifying glass. It can look at a very small piece of human tissue and it can tell you whether or not that human tissue is cancerous or normal. And this is very powerful because during surgery, surgeons need to know exactly which areas of the tissue is cancer and which part is normal. And using the mass spec pen, it can simply touch it to detect if it's cancer or normal, and therefore surgeons can make very exact excisions of the tissue so that the patient has a better chance of survival and a long-term outcome. So thank you.
Thirty seconds to vote. Okay, can we have speaker number 6, please? And go.
Before I begin, I want to ask you guys a question. How many of you have put on sunscreen before? Alright, so all of you might have been in contact with something called titanium dioxide nanoparticle, which is a thing that blocks UV. My name is Leo, and I'm going to talk to you about my research in nanoparticle simulation. So nanotechnology has been a rapidly developing field because material that we think we know do not undergo this or behave the same way we think they do. Iron, the metal, for example, melts at 1500 degrees Celsius, while iron, the nanoparticle, can spontaneously combust in air. So we can make all sorts of nanoparticles. We can vary them, matching different metals together, mix this in different proportions, and we can also have different sizes, 50 atoms small, 1,000 atoms big. And in truth, there are countless configurations, and laboratories cannot go through all of them. We don't have the time, the money, and often the technique to make them. So we go to computers. My field is computational chemistry, where I create and test programs that simulate those hypothetical nanoparticles. We set up the elements, the sizes, and the proportions, and we calculate the physical and chemical properties by figuring out their geometry. Geometry determines everything. It's sort of like the proteins in your bodies, their functions all dictated by their shapes and orientations. It's the same thing for nanoparticles. Turns out that finding the geometry is a difficult task, as the time required to find the geometry increases exponentially as you increase the size of the nanoparticle. So a job can take anywhere from a couple hours to a couple days, and that is on a supercomputer. So I write out modifications like genetic algorithms and test different combinations of parameters to make the program more efficient. That way, we can sift through all the configurations faster, and we can forward that information to the laboratory so that they can filter through and find the configurations of interest. And from that, we can accelerate the R&D process to find those novel nanoparticles and hopefully benefit all of us in our daily lives. Thank you.
Thirty seconds to vote.
Hello. Okay. I have a loud voice. I can't do this or everyone will go deaf. I gotta find
Okay, and ready?
Ready when you are.
Go.
All right, close your eyes. I want you to imagine, if you will, that you're standing in line, next in line to meet Tom Brady, one of the greatest football players of all time. You go up to him, you shake his hand, his grip is surprisingly weak, but as you're shaking his hand, he leans in and says, "Hey, right now as I'm shaking your hand, I am giving you a copy of all of the DNA that makes me an amazing football player. I still retain it too, but now you can go and do whatever I can do." You can open your eyes now. Now, while that sounds like the genesis of perhaps a superhero movie or a comic book, this is something that happens all the time, but not between human beings, between bacteria, which is what I study. Instead of passing around things like awesome football skills or athletic ability, they pass around something a little bit nastier, things like antibiotic resistances, things like toxins, things that help them invade your body and get you sick. My name is Aaron Conrado. I work in the lab of Dr. Brian Davies, and I research the ways in which bacteria are able to take up this foreign DNA, this foreign genetic material, and utilize it. So this obviously is a big issue for public health in two ways. The first way is that it can potentiate the emergence of new pathogens, of new bacteria that can get you sick. It can create bacteria that can get you sick from ones that shouldn't. Have any of you ever heard of like an E. coli outbreak on lettuce or on spinach, affecting your food? That E. coli that can make you sick is not very different from the E. coli that lives in every single one of our guts, which actually loves you. You are its home. It wants to protect you. It wants to keep you safe. The difference between the two is that the E. coli that can get you sick has picked up these extra genes that allow it to do that. The second big problem is that we are running out of antibiotic options to fight these infections that have picked up these resistances. Has anyone in here ever had gonorrhea? I'm just kidding. Don't put your hand up. That's very personal. There are some scientists that predict that within 10 years, if you go to the doctor with a gonorrhea infection, they will say, good luck, because we are out of options to treat you. There are further predictions that predict within 30 years, antibiotic-resistant infections will kill more people than cancer, which is the #1 global killer right now. So in our lab, instead of participating in this arms race where we create an antibiotic and then bacteria become resistant, then we create a better antibiotic and then they become resistant to that one, instead of competing in this infinite war that we will never win, that they will always win, we'd like to kind of sidestep that battle and try some alternative strategies. Maybe we can find ways to stop them from being able to utilize all of this foreign information that they're picking up. Because everything that bacteria pick up is not good. They already have ways to filter out the good and the bad. So if we go back to our Tom Brady analogy, imagine instead you're shaking hands with O.J. Simpson. And he says, hey, I'm going to give you my awesome judgment and my ability to flee from the police in a white Bronco. That's probably not something that you'd want.
Thank you.
Thank you.
30 seconds to vote. Okay, and speaker number 8, you ready to go?
Yeah.
All right, begin.
Cells, like us human beings, speak a language. That language is not called English, Spanish, or French. That language is actually called molecules. So what are molecules? Molecules are a bunch of tiny spheres joined together, and those tiny spheres are called atoms. So how do cells respond to molecules? Well, similarly how humans have ears and eyes to respond to our external stimuli, cells have these organs called receptors that can respond to those molecules. So basically, a molecule can attach to the receptor like a puzzle piece. So our lab is like a designer studio. We design those molecules that can bind to a specific receptor in the brain called the sigma-2 receptor. Okay, so when we design these molecules, we can't just add atoms together freely like Lego. For example, when you're baking a cake, do you add the frosting first? No, you don't. You mix the flour, the eggs, the wheat, you bake it in the oven. Until then, you can't add the frosting. Similarly, we have to add atoms in a certain way. There are rules. Okay, so you might be wondering, what are the recipe steps used to make our cake? Our molecules, sorry. So, let me see. There's Hartwig cross-coupling, alkylation, conjugate addition. Wait, hold on. I think I'm using too much jargon. Let me explain. Alkylation is adding a string of carbon atoms to our molecule, kind of giving our molecule a little tail to play around with. Reductive amination is changing a light bulb. You simply take an atom called oxygen for a molecule and replace it with a slightly bigger atom called nitrogen. And okay, we create a bunch of molecules. Some of our molecules have this really long tail. Some of our molecules have this arm-like structure. The point is, all of our molecules are slightly different, and that's the point. Basically, they bind to receptors slightly differently. You know how when you connect the incorrect puzzle piece, how the rounded tabs still kind of fit, but there's space in between. Same concept. Some of our molecules fit to a receptor very loosely. Some of our molecules fit to the receptor nice and snug. So by creating all these molecules, we can hopefully one day define what the sigma-2 receptor is. We don't know what it really is. It's still a mystery. I'll leave you in suspense. Thank you.
Thirty seconds to vote. Okay, speaker number 9, are you ready?
Yes.
Begin.
My name is Elisa Friedman, and I'd like to take you on a biogeochemist's walk through the forest. So as we walk, we see that there are many components to this system. There are trees, there are animals, maybe we saw a deer, but there's also the soil between under our feet, and there are fungi, there are bacteria, and there are also nutrients like nitrogen and carbon that really make the building blocks of these higher level organisms. And what we know about an ecosystem is that all these parts work together and they integrate to make a balance. But we also know that if we change one of these components, that has a cascading effect over the rest of the parts of the system, all the way down to those building blocks, to that nitrogen, to the carbon. And while we know that, we know that the effects on animals, such as the deer and the trees, but we don't know much about those building blocks. Again, the nitrogen and the carbon. And so my research looks at how the nitrogen cycle especially is affected by these different disturbances, and then also how it bounces back from restoration techniques and how we use a system or manipulate a system to try to bring it back to a native state or a state before a different intervention. And so the way that I look at the nitrogen cycle is that as it moves through different forms in the soil, it transforms. And these different forms can be tracked by using isotopes, which is basically a heavier version of that same nitrogen atom. And these I have inserted nitrogen into the soil, and this soil has, as it goes through, over time it transforms. And so I take a soil sample in the beginning and a soil sample after a certain period of time, and the amount of nitrogen in these different pools can then tell me how it has changed and whether the system as a native state or whether system as it has been disturbed, so in agriculture or in logging, like in the forest, has had any effect. My system especially is in Florida. It's a sandy soil area, and there's been an agricultural plot, a disturbed plot where there's been invasive species, and then a native plot. And so when we have these three different levels of disturbance, we also apply different levels of restoration. Again, there's above ground restoration, so we can focus on plants and taking and removing invasive species, but then there's also below ground problems. So what kind of fungi and microbes live in those areas, and can we seed them? Can we put them back in the system? So these different techniques all interplay and affect the nitrogen cycle. And so when I take a sample at one point and apply these restoration techniques, and then I take another sample at another point, I can get at how these nutrients have changed over time. Once I have these samples, I can basically do a little bit of lab chemistry to put them into a form that I can measure how much there is.
30 seconds to vote. Speaker 10, you ready? Begin.
Leukemia is a cancer of the blood, and in the laboratory of Dr. Lauren Ehrlich, we study a specific type of leukemia called TALL, or T-cell acute lymphoblastic leukemia. This is a cancer of developing T cells, which are part of our immune system. TALL is mainly a pediatric cancer, meaning that it primarily affects children, particularly males. I've even gotten to meet TALL patients, and this cancer really takes a lot out of people. TALL is normally treated with chemotherapy, but one of the problems with chemotherapy is that it's very toxic and has a lot of side effects. Most importantly, a group of TALL patients don't respond to chemotherapy, and for those particular subset of patients, they have a 20% 5-year survival rate, meaning only 1 in 5 will make it another 5 years. Our lab wants to develop a new therapy for TALL. So to do that, we looked at a specific type of immune cell called myeloid cells. My work showed that myeloid cells are present with this TALL tumors in our mouse model. And our lab's previous work showed that if you put TALL cells in a petri dish, they die if they're left on their own. But if you add myeloid cells to that petri dish, they'll grow and survive. I hypothesized that myeloid cells are required for T-ALL to grow and survive in vivo. And to answer this question, I looked at T-ALL in our mouse model, and I found that when you deplete myeloid cells, you eliminate the tumor burden. By killing myeloid cells in these mice, you kill the tumor. I like to think about it with an analogy from my favorite cartoon character, Popeye the Sailor Man. We think that myeloid cells are required for T-ALL to grow and survive, much like spinach is required for Popeye to grow and get big. If you take away the spinach from Popeye, he stays nice and small. If you take away myeloid cells from the T-ALL tumor, then the tumor dies. Our lab thinks that by targeting myeloid cells, we can develop a new therapy for T-ALL. Thank you.
"30 seconds to vote. Speaker 11, you're ready to go?
Yep.
Begin.
Drugs are super important. Not that type of drugs, but like antibiotics and cancer therapies. They've basically enabled the modern way we live. But do you even know how they work? So most drugs target a protein that is essential to the way we live. Take penicillin, for example. It targets bacteria cell walls and stops the protein from building them, so eventually they'll burst and die. Most proteins have a pocket that a drug binds to and stops this essential reaction. So you can think about a protein like a lock, and an antibiotic is like a key that opens the door for death. Simple, right? So then drug discovery is just finding the right key or picking the lock, right? But what about a more complex lock, like a fingerprint scanner? What do? How do you begin there? That's the complexity that's required for most cancer drugs. I'm working on developing a drug for a protein in our bodies, RAS. RAS is a protein that starts signaling in your whole body that tells it to start dividing. So if RAS is malfunctioning, your cell is going to divide uncontrollably and without regulation. This unregulated cell proliferation or growth is commonly referred to as cancer, and RAS malfunction is found in roughly 20% of most cancer types, which is pretty big. Yet, for over 20 years, no drug for RAS has ever been found. Why is this? That's because RAS has a lock system equivalent to the CIA level lock system. It has no drug binding pocket that's obvious for us to use. Instead, it has like a bumpy surface or an interface. So where do you begin? We turn to nature. She always has the answer. There's a related protein that has the same bumpy surface or interface that has a natural inhibitor or a natural drug to stop the function. And what we're investigating is how the drug stays on this surface. And if we can measure and apply that to the cancer-causing RAS system, then we can stop RAS from working and stop your cells from overproliferation, stop cancer. In conclusion, I am cracking the CIA-level lock system that is the cancer-causing RAS system. So hopefully our cancer cells will die. Yay!
"30 seconds to vote. Speaker 12, you ready?
Yes.
Begin.
Good afternoon. I'm Sofia Rojas, and I've been working for a year and a half with a professor in the astronomy department, Steve Finkelstein, and his graduate student. And we have been working on finding the most distant galaxies in the universe that no one else has seen before. So this is very exciting. And now I want to take you on a trip to the past. And I'm going to start with the Big Bang. So we truly believe that the universe started with the Big Bang. And this was a very dense and hot universe. So at this point, all the particles were jumping around and floating with hitting all the other particles. But then the universe starts to expand, and so the temperature drops. And when this happens, then you start forming neutrons, protons, then atoms, and then you start forming stars. And even though you're forming stars, the universe is actually looking dark. Why is this? Because you don't have enough energy to see light. But then there's some point, we don't know when galaxies start to form with these very massive stars called Lyman alpha, that is the most energetic wavelength you could see of hydrogen. And at this point, the universe is illuminated. And we call this the epoch of reionization. So what I'm looking at in my research is finding these galaxies that cause the reionization of the universe. So how do I do this? Well, we have the best telescope so far, I believe, the Hubble Space Telescope. And this telescope takes imaging in random spots in the sky, all over, so that you have a good statistics of the number density of galaxies at a specific epoch of the universe. So in this case, the epoch of reionization. So basically, I look at the images in 5 different filters. And the way to think about these filters is that you have a bluest filter, and then you go to the 5th one that is at a redder wavelength. And what I'm trying to see is when I start to see a point of light between these 5 filters. And if it's in the reddest filter, that means that I'm probably looking at this galaxy that is very far away, because again, the universe is expanding, so everything is looking more towards the reddest wavelengths. Unfortunately, I have worked on this project for a while, and we've already found 3 of these galaxies that no one had seen before, and it's very interesting, but these are only galaxy candidates, because the way you can confirm this galaxy is if you look at the spectra of the galaxy and actually measure that Lyman alpha that I mentioned at the beginning. So that is another step that I will have to take, but for now, we have some good candidates. Thank you.
"30 seconds to vote." Speaker number 13, you ready?
Sure. Why not?
Begin.
Hey, y'all. Hope you're having a great day today. Have you ever been there texting your friend saying, "I'm sorry, I can't make dinner tonight. I have to reschedule." Your friend texts back, "Okay." Great, you're relieved. Your friend's not mad at you. You can reschedule. Everything's fine. But then you show this text message to another one of your friends, and they say, "Are you crazy? Your friend's totally pissed at you. They wrote back, okay." What we have here is 2 people taking the same bits of information, the same text message, the same context, but they're interpreting it in 2 very different ways. And that's at the heart of what I do. My research focus is that I look at how people make decisions in public scientific controversies. So things like climate change, GM foods, or genetically modified foods, or in really particular, vaccines. 99% of scientists agree that mankind is responsible for climate change. Yet only half of the public agrees with this. Why? Part of my work is developing a new theory to explain how people interpret scientific information and how they come to a certain decision about a scientific issue, particularly when it conflicts with the scientific mainstream. If we look at how we make decisions about controversial topics, so things like gun control or abortion, we often use our values, things you might consider your morals, your ethics, things that are important to you, to be this guiding rod in how you interpret this information and how you make a decision. Now let's take a look at how we make decisions about scientific issues. For most of us, we actually look to experts. So things like scientists, doctors. If I were to ask you to make a decision about how you feel about artificial intelligence, you might look to Elon Musk or Bill Gates. I think the way we make decisions about controversial topics is now actually how we're making decisions about scientific topics. So the way we make decisions using values on gun control is the way we're making decisions about vaccines. I think values play a much larger role than we've ever anticipated in how we make decisions about scientific topics and how we interpret scientific information. Particularly if you think about how values influence the way we vote, what we believe, and in some cases, how we interpret text messages from our friends. Thanks.
Thirty seconds to vote. Speaker 14, are you ready?
Yes.
Begin.
All right. By the time I'm done with this presentation, unfortunately, 35 mothers, fathers, and children will pass away. The disease that will make them pass away is very commonly known, and it's cancer. Now, cancer is a disease in which there's uncontrolled cell growth in our body, and that uncontrolled cell growth can spread to other parts of the body and cause death. Now, in my group, we are looking at how we can use the immune system to fight cancer. Now, we all know that the immune system can fight off bacterial infections, viruses, but it turns out that immune cells can also recognize and fight cancer cells. Now, you may be wondering, how can they detect cancer cells? Well, it turns out that when cancer cells are lurking around in our body, other immune cells attach to main immune cells and kind of form a handshake. Now, this handshake can help activate these immune cells and tell them to fight, that there is cancer here, let's fight it. Now, there are other types of handshakes where the other immune cell says that the coast is clear. And this tells the main immune cells to not fight this cancer. Now, unfortunately, cancer takes advantage of this handshake and actually tells other cancer cells, tells other immune cells to not actually actively fight the cancer. So in effect, it evades our body. Now, what we are trying to do is to look at a subset of immune cells called T cells and try to see if they are being taken advantage of by these cancer cells. And hopefully, through our research, we will find a way to block this inhibitory effect by T cells and prevent cancer from hiding from our soldiers. Thank you.
Thirty seconds to vote. Okay, here's what's going to happen now. We're going to ask you to pass your ballots to our volunteers over here. Volunteers, raise your hands, please. All right, pass your ballots to the volunteers. If you have an orange ballot, pass to the volunteers, and then there will be a period of tabulation, okay? And let's see. Projector's on. And we're going to ask you to take a bit of a survey while that is happening. And just kind of listen to some tunes that I'm going to pull up here. And then, once we have finished the tabulation, we will announce who the winners are. Judges, if you have a green ballot, pass that back to me, please. Oh, okay. All righty.
Hi, this is Greg Popovich of the San Antonio Spurs, and you're listening to 91.7 FM, your home for jazz, KRTU San Antonio.
Support for KRTU comes from the San Antonio Current, presenting the 12th Annual San Antonio Beer Festival.
90s recording of Gene Harris, a pianist there on a Kenny Dorham composition. That was Blue Bossa. That's how we start off the sixth.
Okay, guess what? All the arithmetic has been done, and all the answers are known. Okay, so I would like to. We're going to start with the audience award, but first I would like to introduce our judge, Christine Sinatra, the director of communications from the College of Natural Sciences and an excellent communicator, to say a few words.
Thank you. I sprung on Roxanne that I just wanted to speak really quickly. I know a lot of you are from natural sciences, and even if you're not from natural sciences, I just want to say how much I appreciate every single one of you for coming up here and doing this and getting in front of everyone. I was wowed. I didn't come in necessarily with super high expectations, but But I have my cards here. I'm the communications director. I love to tell stories, and if you'll grab a card on your way out, however it turns out up here, and please be sure to send me an email if things are happening with your research group. Everything I heard sounds like a great story to me. So anyway, congratulations to all of you.
Okay. Don't go yet. And Christine is going to announce the number of the speaker who has won the audience award. And when that person comes up, please tell us your name and your department and your status. Are you undergrad, master's student, doctoral student? What's up with you? And your department, I think I said that. Okay, so here we go. It is lucky number.
She just told me what it was. So I actually didn't know the announcement so much faster than you. But it's number 3. It's number
I think she had to leave. Lucky number 3 had to leave.
Oh, so do we know the answer to who that was?
We do know the answer to who that was. Lucky number 3 is Savannah Troy. Okay. All right, so she talked about the sparrows. All right, and now I would like to introduce Dr. Rob Millman. He's a colleague that I met at the Alda Center for Science Communication at a Summer Institute in 2014, and it's been a pleasure to work with him. He's affiliated with the Moody College of Communications Center for Health Communications, and he's affiliated with Austin Radiological Association, which supports this contest and underwrites the prizes. And he's here to announce the Judges Award.
Thanks, Roxanne. And you don't know how much effort goes into this, but Roxanne does a tremendous job. So let's have a round of applause for Roxanne. And ARA is happy to sponsor this. I'm a radiologist by training, and I do a lot of health communication trainings as well. Some of you have come to those trainings. And without further ado, the Judges Award goes to number 11. Woo!
Hello, my name is Lisa Strong. I'm a senior biochem major in the Department of Molecular Biology. My work is in the Department of Chemistry with Dr. Lauren Webb, and I'm intending to go to grad school, hopefully. Okay.
So I would also mention, I believe that you were an alternate and you moved up to contestant as other people were unable to attend this evening. So if you're interested in science and plain English, and you're not the only alternate who spoke today who moved up, right? Someone else. So show up, okay? If you're interested in doing this next year, show up. It could be that other people have other commitments that come up and they can't make it, and you might move up to win all the things. All right, that kind of concludes what's going on, except that we really want you to take this survey here and let us know what you think about this, how we can improve it, things like that. We really, really want you to eat those snacks so I don't have to take them back to my office. So there's that. And just stick around. In the spring, the UT Science Communication Interest Group has a signature event that's called Research Speed Dating. It's a similar 3 minutes to talk about your work thing, but you sit across the table from someone who is in a completely different field. They give you feedback, so you'll get actual feedback in that event. And then, you know, if you've done the speed dating thing, you know, you move down a chair, and except this time you flip and you're the listener giving feedback to the person who's across from you. We used to call it science speed dating, but last year we were fortunate enough to get together with the folks from Not Even Past, which is an initiative out of the history department that's public scholarship, right? Public engagement, whether it's science, whether it's whatever scholarship. And so we had a lot of folks from humanities and from fine arts who came to participate. So now it's grown from science speed dating to research speed dating. It's usually in February, and be looking for it. And thanks a lot. Eat the snacks.